A general principle of biocompatible dentistry would state that everything we implant or leave in and around a tooth represents a systemic exposure whose impact must be accounted for and considered. The good news is there is an extensive program of testing for materials newly introduced to the market. The ISO 10993 standard and its American version, the FDA's Blue Book Memorandum G95-1, outline these requirements. The problem is that in the real world of the dental marketplace, this thorough testing regime is almost always bypassed by the FDA's grandfather clause, Section 501K of the FDA statute of 1976. As well, laboratory benchtop biocompatibility testing doesn't account for variations in patients' individual responses to artificial materials.
Perhaps in an attempt to improve the status quo, in 2016, the United States Food and Drug Administration, FDA, published a document entitled, quote, Use of International Standard ISO 10993-1, Biological Evaluation of Medical Devices, Part 1, Evaluation and Testing Within a Risk Management Process, end quote. The purpose of this publication was to provide non-binding recommendations and to offer the current thinking of the FDA on the topic of risk of medical devices. The source for this is the U.S. Department of Health and Human Services, Food and Drug Administration Center for Devices and Radiological Health, 2016. An updated version of the ISO 10993-1 was subsequently published in August 2018. It contained some changes in regard to medical device testing, and it was stated that the primary aim of that document was, quote, protection of humans from potential biological risks arising from the use of medical devices, end quote. The source of this is ISO/TC 194, Biological and Clinical Evaluation of Medical Devices, 2018. However, a company that offers support for medical device testing, including, quote, rationales for the selection or waiving tests, end quote, explained of the new version ISO 10993-1 published in 2018. Quote, in the new version of the standard, not all biological effects should be assessed by biological testing. Some tests can now be waived if there is sound rationale to support the decision, end quote. The source for this is Biological Evaluation of Medical Devices as an Essential Part of the Risk Management Process, Updates, ISO 10993-1, 2018. At the 2019 8th European Workshop on Visual Information Processing, researchers openly stated that the standard ISO 10993 is, quote, not intended to provide a fixed set of testing methods, including acceptance criteria, end quote, and that the final opinion is on the manufacturer. These types of comments have led many to consider these regulations as more favorable to industry's profits than protective of consumers' health. The source for this is M. Augustinic et al., Biocompatibility of Medical Devices and Their Risks, the 2019 8th European Workshop. The situation has been gaining attention from concerned citizens and even investigative reporters. For example, the International Consortium of Investigative Journalists shared medical implant stories from their readers in November 2019. They collected more than 3,500 responses from people affected by the medical device industry in 50 countries, bringing to life the personal cost of implants that fail. To date, they are still running news stories as part of their implant files. Source for this is Wilson Chapman A. et al., International Consortium of Investigative Journalists Implant Files, November 2019. The image is of Christina Chung, ICIJ, and reproduced with permission from the International Consortium of Investigative Journalists. While industry has continued to profit from the new testing guidance, injured consumers have continued to fight back. Most notably, public pressure was put on the FDA about adverse health effects from dental amalgam fillings, as well as the metal-containing birth control device, Essure, which was eventually ended for distribution in the USA on December 31st, 2018. These concerns and more finally led the FDA to host a meeting of the Immunology Devices Panel about dental amalgam and metal implants on November 13th and 14th, 2019. Prior to the panel meeting, the FDA had shared two new documents, one being a literature review on dental amalgam and the other being a review of scientific information related to metals and their uses in implants. Some IAOMT members wrote responses to the FDA highlighting risks and studies overlooked in their reviews, and Dr. Jack Call, the executive chairperson of the IAOMT Board of Directors, spoke at the Immunology Devices panel meeting to share pertinent research and express concerns. The following is an excerpt from his presentation.
Good morning. My name is Jack Call. I have no financial interest or conflict. I've paid for my travel and hotel costs. For 42 years, I've practiced general dentistry. 36 years ago, when I learned that mercury escaped from amalgam fillings, I immediately stopped placing them. I serve as the executive chairman of the board of directors of the International Academy of Oral Medicine and Toxicology, a nonprofit organization. This is a volunteer position. I'm here representing our over 1,000 international dentist members. I'd like to direct the focus of inquiry to mercury vapor, which was not acknowledged yesterday as to its equal ability to cross the blood-brain barrier just as readily as methylmercury does. Yesterday it was noted that mercury vapor is the predominant form of mercury exposure from dental amalgam, as 80% of this vapor is absorbed through the lungs during inhalation. This mercury vapor off-gasses continuously from the amalgam surfaces. The rate of release increases due to any temperature increase, either from hot food and beverage consumption or friction from eating, bruxism, and toothbrushing. In a minute, I'll show a video that was part of a study published in the journal Neuro Report in 2001. It summarizes in a visual way the methodology and results of this study, which can help us understand the pathophysiology of mercury. The following is a quote from the paper. The mercury concentrations to which these neurons were exposed, 0.1 micromolar, were of the same order of magnitude as mercury levels reported in human and animal studies after chronic exposure to mercury vapor. The actual mercury concentration present in our neuronal cultures was indeed lower than the 0.1 micromolar because of a dilution effect in the culture media.
How mercury causes brain neuron degeneration. Mercury has long been known to be a potent neurotoxic substance, whether it is inhaled or consumed in the diet as a food contaminant. Over the past 15 years, medical research laboratories have established that dental amalgam tooth fillings are a major contributor to mercury body burden. In 1997, a team of research scientists demonstrated that mercury vapor inhalation by animals produced a molecular lesion in brain protein metabolism, which was similar to a lesion seen in 80% of Alzheimer-diseased brains. Recently completed experiments by scientists at the University of Calgary's Faculty of Medicine now reveal, with direct visual evidence from brain neuron tissue cultures, how mercury ions actually alter the cell membrane structure of developing neurons. To better understand mercury's effect on the brain, let us first illustrate what brain neurons look like and how they grow. In this animation, we see 3 brain neurons growing in a tissue culture, each with a central cell body and numerous neurite processes. At the end of each neurite is a growth cone where structural proteins are assembled to form the cell membrane. 2 principal proteins involved in growth cone function are actin, which is responsible for the pulsating motion seen here, and tubulin, a major structural component of the neurite membrane. During normal cell growth, tubulin molecules link together end-to-end to form microtubules, which surround neurofibrils, another structural protein component of the neuronal axon. Shown here is the neurite of a live neuron isolated from snail brain tissue, displaying linear growth due to growth cone activity. It is important to note that growth cones in all animal species, ranging from snails to humans, have identical structural and behavioral characteristics and use proteins of virtually identical composition. In this experiment, Neurons also isolated from snail brain tissue were grown in culture for several days, after which very low concentrations of mercury were added to the culture medium for 20 minutes. Over the next 30 minutes, the neurite membrane underwent rapid degeneration, leaving behind the denuded neurofibrils seen here. In contrast, other heavy metals added at this same concentration, such as aluminum, lead, cadmium, and manganese, did not produce this effect. To understand how mercury causes this degeneration, let us return to our illustration. As mentioned before, tubulin proteins link together during normal cell growth to form the microtubules which support the neurite structure. When mercury ions are introduced into the culture medium, they infiltrate the cell and bind themselves to newly synthesized tubulin molecules. More specifically, the mercury ions attach themselves to the binding site reserved for guanosine triphosphate, or GTP, on the beta subunit of the affected tubulin molecules. Since bound GTP normally provides the energy which allows tubulin molecules to attach to one another, mercury ions bound to these sites prevent tubulin proteins from linking together. Consequently, the neurite's microtubules begin to disassemble into free tubulin molecules, leaving the neurite stripped of its supporting structure. Ultimately, both the developing neurite and its growth cone collapse, and some denuded neurofibrils form aggregates or tangles, as depicted here. Shown here is a neurite growth cone stained specifically for tubulin and actin, before and after mercury exposure. Note that the mercury has caused disintegration of tubulin microtubule structure. These new findings reveal important visual evidence as to how mercury causes neurodegeneration. More importantly, this study provides the first direct evidence that low-level mercury exposure is indeed a precipitating factor that can initiate this neurodegenerative process within the brain.
But alas, I have fallen into the same trap the gentleman from Consumer for Dental Choice scolded us about yesterday, in continually arguing about scientific studies, their imperfections, and all the information gaps. Yesterday, I heard the following phrases: individual variability, underlying assumptions, conflicting results, and somewhat imperfect. Even though intellectually we might all agree that a perfect study on amalgam safety will never occur, the FDA's behavior sure doesn't reflect that. They'll keep hoping to find one forever. Over the years, we've been told that the FDA does not consider the precautionary principle, nor additive or synergistic considerations. For example, when there is concurrent exposure to both lead and mercury, even though it is well documented that both together are more toxic than one by itself. Let's face it, what has been occurring the last 3 decades is a racket. The institutional inertia for a grandfathered substance, mercury fillings, the alleged safety of which is perpetuated by a trade organization, the American Dental Association, and which is then tolerated by the government body responsible for protecting the public, the FDA. I've got one word for it: reprehensible. My call to action for the FDA is to apply the precautionary principle and start now by restricting the use of amalgam in children, pregnant women, and those with kidney and neurodegenerative diseases. In conclusion, relative to some comments made yesterday, I've been placing composites which are BPA-free for years now. Thank goodness the dental profession has very good alternative materials. Additionally, I was the dental director of a federally funded community health center serving indigent Medicaid populations for 25 years. I figured out how to utilize rubber dam to provide high-quality adhesive dentistry without amalgam, just like dentists in the countries where amalgam use has been eliminated or heavily reduced.
Unfortunately, the issue of metal implants and dental amalgam fillings contributing to adverse health effects was left unresolved at the 2019 Immunology Devices Panel meeting. A reporter from MedTech Dive explained, quote, aside from recommending FDA implement more transparent labeling, the meeting ended with few clearly laid out action items, end quote. The source, Amrishal, FDA advisors, metal implants need beefed up ingredients label. MedTech Dive, November 15th, 2019. The FDA now states on their website, quote, The science around immune responses to materials and medical devices is expanding, and furthering this science to improve patient treatment options and outcomes is a priority for the FDA. To better understand how a patient responds to materials used in medical device implants and harness that information to improve the safety of devices in patients, the FDA is working to engage the public, in particular, scientists, patients, and healthcare providers, and industry stakeholders to determine the current state of the science, critical gaps in the existing science, and what approaches the FDA should consider, end quote. The FDA also states, quote, the FDA is interested in stakeholders' perspectives on our recent review and any other new information to help inform our understanding of the current benefit-risk profile of dental amalgam, end quote. The source for this is the U.S. Department of Health and Human Services, FDA, Metals Used in Medical Devices, FDA website. Access July 13th, 2020. Clearly, this is an issue in which we, as biological dentists, should continue to closely monitor. Future regulatory deliberations are inevitable based on the FDA's current stance. Meanwhile, millions of patients around the globe have these materials placed inside their bodies with little to no regard for potential side effects and variants in their biochemical and immunological responses.